Hi, my name is Jingler Antunes and I'm a postdoctoral research associate at Rice University. I'm also a fellow of the Computational Cancer Biology Training Program of the Gulf Coast Consortia with funding from the Cancer Prevention and Research Institute of Texas. This is a two-part video tutorial on the use of HLA-Arena. In the first part, I'll give you some background on the importance of modeling peptide-HLA complexes and on the methodologies behind some of the tools included in the HLA-Arena environment. On the second part, I will demonstrate how to download and execute the HLA-Arena workflows. To give you some context, HLA stands for Human Leukocyte Antigen, which refers to this family of receptors that are capable of binding peptides derived from intracellular proteins and displaying them at the cell surface. By doing so, they expose this intracellular content for recognition by T-cell lymphocytes. T-cells will not recognize peptides derived from self-proteins from healthy tissue, but they will recognize peptides displayed at the surface of tumor cells, for instance, or at the surface of infected cells. The recognition of tumor-associated peptides at the surface of tumor cells will trigger T-cell activation and the elimination of the tumor. Therefore, predicting which peptides are capable of being displayed by HLAs at the surface of tumor cells is of key importance for vaccine development and cancer immunotherapy efforts. And many of the consequences or the features of this recognition in terms of the affinity and specificity of these interactions are driven by structural features. So predicting the structure and analyzing the structure of these peptide-HLA complexes is very important for these cancer research applications. Here you can see an overview of the structure of the HLA receptor. It's an heterodimer. In blue you have the heavy chain, in green you have the light chain, and you can see the peptide in the binding site. So the structure of the HLA receptor is very conserved in terms of folding, although there are many variants, more than 19,000 alleles in the human population, but the overall folding is quite conserved, so you can apply homology modeling to predict different structures in addition to the structures that were already determined by experimental methods. The reference computational method to predict the structure of a protein-ligand complex is molecular docking, and there are many different tools that are routinely used for drug discovery. These tools, however, best perform for ligands with up to 12, maybe 20 flexible bonds or degrees of freedom. And the challenge here is that peptide ligands can have way more than 30, 40, or even 50 flexible bonds, being too large and too flexible for conventional docking methods. As an alternative, our group has developed an incremental approach called Dink. Instead of docking the entire ligand at once and exploring all the flexible bonds of the ligand, Dink selects a small fragment with, let's say, up to 6 degrees of freedom or 6 active flexible bonds, here depicted in green, and runs the molecular docking to find the best binding mode for that initial fragment. After this first round, Dink selects a number of additional atoms to be added to the initial fragment, here displayed in red, and selects a new subset of bonds to be explored in a new round of molecular docking. This process of docking and growing is repeated incrementally up to the point in which the entire ligand is predicted in a bound conformation to the receptor. Dink is in fact a meta-docking approach in the sense that it relies on conventional docking tools to perform the sampling and the scoring of each of these fragments while managing the parallelization and the incremental process. To learn more about Dink, you can check our recent publication on BMC Molecular and Cell Biology, in which we describe the latest version of the algorithm and we describe its performance in reference data sets of large ligands, including mostly peptides. However, as discussed in the HLA-Arena paper, more consistent results are obtained when using a higher number of threads, and the prediction of a single complex can take more than an hour depending on computational resources and the length of the peptide being predicted. Therefore, Dink can be used for geometry prediction for different HLA receptors and for peptides with different lengths, but is not suitable for large-scale analysis or virtual screening. The search for scalable solutions led us to implement APEGEN, which is the anchored peptide MHC ensemble generator. APEGEN is based on the assumption that peptides displayed by class 1 HLA receptors are spatially constrained to very specific positions in the binding cleft. So to perform a new prediction, APEGEN actually uses templates for these specific positions from experimentally determined crystal structures. These templates are used to position the backbone of the termini amino acids for the new peptide we want to model. And by doing that, we transform this problem into a loop closure problem, since we need to fill this gap. To do that, we use a fast geometrical approach called random coordinate descent that will generate a number of valid backbone conformations connecting the two termini residues in this peptide. Once we have this ensemble of backbone conformations, we then proceed with the full atom side chain reconstruction for each one of these backbones according to the sequence of the peptide we want to model. During this process, we will explore the flexibility not only of the side chains of the peptide, but also of the side chains of the receptor. And this process is done with ISMINA, which is also a docking tool. This step will create an ensemble of valid low energy conformations for the peptides bound to the HLA receptor. And we can actually refine the results by selecting the best conformation or the best complex here and using as input for a new round of refinement. Note that this is a very efficient procedure, taking about 2 minutes to model a single complex in a regular desktop computer, and we have demonstrated that the generated ensembles include near-native conformations that are close to those observed in crystal structures. Now we have created HLA-Arena, which is a computational environment that integrates APEGEN and Dink with other tools for molecular modeling and analysis. All these tools were packaged into a Docker image, facilitating the installation and use in different operational systems. We have also created a number of workflows through Jupyter Notebooks to facilitate the use of HLA-Arena for different applications, which include geometry prediction, binding affinity prediction, and virtual screening based on structural features. Users can customize available workflows according to their needs and also create completely new workflows using Jupyter Notebooks and using the functions provided by the HLA-Arena Python module. For more details on how to use and how to customize these workflows, please check the second part of this video tutorial.